Hello friends, welcome back to my channel Medicine 101. Today we'll talk about insulin synthesis and secretions. Okay, it's the aspect from the physiology as far as you can see in terms of the pharmacology also. Okay, we'll talk about all these things in details. Now, starting the insulin secretion and synthesis, let's talk about from where the insulin will come. So it's from the very basic knowledge we know that insulin will be secreted by the what pancreatic beta cells. So before that, let's draw a little bit of the pancreas over here. Okay, so let me draw. It's not so correct, but for understanding purpose I'm just drawing like this. Okay, now see, if we just see within the pancreas is the both type of the things we can see over here. It acts as exocrine as well as it acts as endocrine, right? Now see, whatever the exocrine function we will see in this case, is exocrine function we will see in this pancreas will get the different types of acinar cells, acini. Just like this, if I show you as so, these acinar cells around the acini, there will be acinar cells just like this. Secret the pancreatic what? Pancreatic enzymes or pancreatic juice just like this, and we will see they will form a duct over here, pancreatic duct, and from the duct cells we will get what secretion of the bicarbonate. So from here we will get secretion of the bicarbonate. Then this bicarbonate will get mixed up with the what? With the pancreatic juice. After that, through the pancreatic duct, it will be just open where within the duodenum. So it will come over within the duodenum. Okay, this is for digestion purpose. Now, what about the endocrine functions? What do we know in terms of the endocrine? Now, if you say in terms of the endocrine, around the 99% cells of the pancreas, that is acinar cells. Now, approx 1% of the cells, that is what for the endocrine functions. Okay, that means we'll get what? Islets of Langerhans. Islets of Langerhans cells. Okay. Now see, if I just show you around 1% over here, okay. Now if I want to show you a single piece of the islets over here through the large diagram, let me show this one. Just like this, okay. So for these things, okay, within the islets of Langerhans, we will see that beta cells will be within the center. The beta cells that will be within the center, just like this. Around the beta cells will get alpha cells. Around the beta cells will get the alpha cells. Okay. And in between the alpha and beta cells will get the delta cells over here as well. So we'll get also the delta cells. And we can also get the delta cells, some portions of here also. Now we'll talk about the different types of the function of the cells over here. And we'll also get the pancreatic polypeptide cells. So we'll talk about these things later, okay? Now, if we just talk about it in terms of the beta cell first, see what are the things of the beta cells do we know? The pancreatic beta cells that can secrete what? Insulin. Along with the insulin, we will see they can secrete also peptide C or C peptide. It's like, okay, let's write it over C peptide. Along with that, they can also secrete what? Amylin. Now, what is amylin C? If we see in terms of the type 2 diabetes mellitus patients, in these cases, we will see there is a chances for the deposition of the amyloid. Okay, you know the amyloid, I think you have heard this term. So in these cases, the amyloid that will be made up of this amylin protein. So in terms of the diabetes 2 mellitus patients, we can see the presence of amylin. Okay, so let's write it over here. In terms of type 2 diabetes mellitus patients, we can get the amylin over here. Okay. And we'll also get in terms of the beta cells, we'll get little amount of the proinsulin. We'll get the little amount of the proinsulin. And okay, beta cells done. Now if you talk about in terms of the alpha cells, what are the functions? They are responsible for secretion the glucagon hormone. Glucagon. Okay. So when they will be secreted under the stress, that means when you have hypoglycemic conditions. During hypoglycemic conditions, the blood sugar will be less. Okay. And whatever these things, the insulin will be secreted whenever the blood sugar will get increased, just to maintain the blood sugar levels, to come back to the normal blood sugar level, the insulin will be secreted. And what about the delta cells? These delta cells suppress the both insulin, the beta cells as well as the alpha cells. Okay, so it suppress alpha and beta cells both. We can see in the better word that it maintains the secretions of alpha and beta cells. That means it makes the balance in between the secretions of insulin and glucagon. Okay, this other thing we'll see over here. Now, if you just talk about a little bit modifications of the insulin, first we'll get the insulin in which form. Then, okay. See, whenever the insulin will be produced, okay, let's write it over here. For example, whenever the insulin will be produced in the initial stage, it will be just it will be named as preproinsulin. Preproinsulin. Okay, I'll talk about how these things are produced. First, before that, I'm just saying a little bit of them. So, if you see the preproinsulin, they will be looking just like this. Looking just like this. Whatever the segments will get over here, see the first segments of this thing will get, this segment is known as what? This from this portion to this portion, this is known as initial segment. Initial segment. Or we can say another name for signal sequence. Signal sequence. Then we will get the portions from, for example, from here to here. It is known as B chain. This is B chain. And the regions, for example, will get from these regions to these regions. It's looking like C shape. Okay, so it is known as C chain. And the portions that left over here, that is known as what? A chain. Known as A chain. Okay. So at first we'll get the preproinsulin. After that we'll get the modifications where we'll see the initial segment that will be removed. If the initial segment is removed from here, then what is left over here? Only the B chain, C chain, and A chain. We'll get over here. B chain, that means let me show over here. B chain, C chain, and A chain. Just like this. And of course we'll get over here the disulfide bond. We'll get the disulfide bond in between the B chain and A chain, and on disulfide bond we'll get intra A chain. Okay, just like this. So we'll get these two type of the disulfide bond over here, and we'll also this the disulfide bond. Now see, in these cases, now the segment what only it is left over with B chain, C chain, and A chain. Now it is known as what? Proinsulin, because the pre part, that means the initial segment has been removed. Now we can call it as what? Proinsulin. It will be known as proinsulin. Okay. After that, we will see that the next modifications we will get over here. Here we will see this C segment, that means the C chain, that will be removed. The next modification we will get the C chain, that will be removed. Now, if the C chain is removed, then whatever the modifications we will get, only A chain and B chain will be removed. Only A chain and what? This is the B chain, this is the A chain, that will be removed over here, and we will see the disulfide one in between them, just like this one. So now this is known as active insulin. So we'll get over here active insulin over here we'll get. Along with that we'll get what? The C peptide. Is the what? C peptide. C peptide. How many insulin molecules will be produced? The same amount of the C peptide we'll get also over here. Okay. Now we'll talk about how we are just synthesizing this what insulin things over here. Okay. So for these things, let's talk about over here. Now, for example, I'm just considering the beta cells over here. Just like this, okay. Now see the chromosomes, chromosome number 11 in this. Okay, here over. Now there are certain types of the genes. Okay, whenever these genes will be activated, there are certain types of the genes. First genes, okay, this will be activated. After that, productions what? They will produce the different types of the mRNA. First, they will produce the mRNA. Now, as we know that what insulin is a peptide hormone. Peptide hormone means definitely there will be productions of the proteins, the amino acids. So for these things, we'll see the translations process, transcription process. So at first, we'll see the transcription. So in the DNA segments of the beta cells, we'll see over here. At first, we'll see the process of transcription from this gene. And whenever the transcription will be started, we'll getting the mRNA. After that, we'll get mRNA will come out of the nucleus. Now the mRNA will be converted into what? Proteins. That means the amino acids. How the mRNA will come out? After that, it will sit where? Within the ribosomes. So if you see there is the ribosomes, okay, the mRNA will come over here, just like this. and it will produce the different types of the amino acids, and the amino acids, they will get connected together. Okay, they will form the peptide chain just like this. Okay, now we'll see these ribosomes, they will just bring the what, this peptide segment where within the endoplasmic reticulum. Okay, now for example is the endoplasmic reticulum. Let me show just like this over here. Okay, now see, at first they will just bring this segment, whole segment from here, these regions. Okay, and these ribosomes have come over here just like this. See, and they are sort synthesizing, they are just sending the segments, the peptide segment just like this. For example, first it will produce what, the initial segment. At first we will get the initial segment that will be produced. So we are getting the initial segment over here. Okay, this is lipid soluble, this one. So it will cross easily the endoplasmic reticulum. So definitely it will be easier for the initial segment to come within the what, endoplasmic reticulum. As the ribosomes will get attached over here, we will get what, this will be known as rough endoplasmic reticulum. Whenever the ribosomes will get attached with the endoplasmic reticulum, it will be known as rough endoplasmic reticulum. Now we will get just like this there. Then we will get the different types of the ribosomes will get attached, and they will just send the different types of the segments. For example, first the initial segment, they are getting the A chain, they are getting the B chain, they are getting the C chains, just like this. After that we will see there will be formation of what, there will be formations of the insulin, just like this. We will get what, these segments will get connected with each other, and we will get over here that the preproinsulin, this one. Now there is the presence of one type of the enzyme within the what, endoplasmic reticulum, that is known as endonuclease. That is known as endopeptidases. Known as endopeptidases, yes. Because the peptide bonds that will get break down. So at first the endopeptidases, what they will do, they will just remove the what, the initial segment from here. Okay. So after that, what we will see over here, what will be left over here? Left portions will see only over here that the B chain, C chain and A chain, and in between them what, the disulfide bond, just like this. Okay. Then we will see this thing just like this. Let me show over here. This thing will be what, get attached with the membrane of the endoplasmic reticulum, just like this. Now we will see whatever thing over here. This thing, that means now it will be known as what, proinsulin. The proinsulin, it will be over here. After that, what we will see, there is the formation of the vesicle within the endoplasmic reticulum, and whenever the vesicle will be formed, after that we will see within the vesicles of this part. Okay, what we will see, the proinsulin. We will see the proinsulin. That means what, there will be also left some C peptide, just like this. Actually, somehow the C peptide that will be removed, but there is also presence of one type of the enzyme, just like the endopeptidase, where there will be also endopeptidase within the Golgi apparatus. Okay, at first it will see within the vesicles. After that, it will be sent within the Golgi apparatus. And whenever it will come within the Golgi apparatus, see, we will get over here only A chain, B chain, disulfide bond, and the C peptide. We will get all these things. Okay, because within the Golgi apparatus, the structures of the proinsulin, from here the C chain will be removed by the action of the what, endopeptidase. There is another type of the endopeptidase that is present within the Golgi apparatus, that will cut down the what, C peptide from the proinsulin. Okay, now we will get only this form. After that, this thing, okay, let me increase size of it. Okay. After that, we will see this Golgi apparatus, it will produce different types of the vesicles after the processing. Now within the Golgi apparatus, we will see there will be stored of insulin. There will be stored of insulin. The C peptide. Now the insulin is getting where, after synthesizing, where it is getting stored within the pancreatic beta cells. Now how the insulin will be secreted? Let's talk about that thing. So we will talk about right now how the insulin is getting secreted over here. Now it's easy also. Now for example, I am just drawing a pancreatic beta cells over here. What do we know? When the insulin that will get secreted, basically whenever in the feeding state, okay, during feeding state. So feeding state. During feeding state, what we are getting? For example, we are getting from the GIT word, from the GIT word, the glucose that will come where within the circulation, and from the circulation we will go to the different types of the tissues. Okay. Now see, what type of the thing we'll see. For example, these glucose molecules, we are getting the glucose molecules, and there is the presence of one type of the transporters who are within the pancreatic beta cells, that is known as GLUT4. So let me just 12 first receptor just like this. Anyway, so this is known as GLUT4. What is the function of the GLUT4? There is one portion receptors within the GLUT4 that is available for glucose. Now it will be available for glucose. Whenever the glucose will get attached over here, we will see this will flip in. That means the glucose molecule that will be taken where within the pancreatic beta cells. Okay. Now we are getting the pancreatic beta cells where within the within the glucose you are getting within the pancreatic beta cells. After that we get over here, there is presence of one type of the enzyme that is known as glucokinase. So we'll see whatever thing we'll see further, the glucokinase. So this glucokinase that will convert the glucose into glucose 6-phosphate. That means it will add one what charged particles along with the glucose. Because whenever the glucose is coming within the cells, pancreatic beta cells, there is a chances for the glucose to go back from the cells to the again outside of the cell. If we just add one charged molecule over here, because we know that the charged molecules they cannot cross the membrane, so the glucose there is no chance for the glucose to go outside of the cell. So glucose will get glucose 6-phosphate. Then after that we will get the further stage. So the glycolysis will get ultimately what pyruvic acid. We will get pyruvic acid. After that we will see this will go over within the TCA cycle. That will go within the TCA cycle, where we will see they will produce what? In these cases we will see the ADP that will get converted into ATP. The ADP that will convert into ATP. The amount of the ATP that will be more. Okay, whenever the glucose is coming. Now see, there is presence of one type of the channel that is known as potassium channel who are within the pancreatic beta cells. Okay, so see, for example, is the is the potassium channels. Now there are certain factors that stimulate the potassium channel. If you see the potassium channel, just like this, see these structures. Okay, just like this. Now within the potassium channel, we will see there is the portions where ADP can bind. These are the portions where ADP can bind. Now whenever the glucose is not coming where within the cells, as a result, we will see there is what? The amount of ADP will be more, the amount of ATP will be less. So whenever the ATP will get bind with this potassium channel over here, okay, with the potassium channel over here, we will see these portions will be left. That means the whatever the intracellular potassium is over here, this all the potassium they will go out of the cell. They will go outside of the cells. So there will be what? In efflux of the potassium. There will be efflux of the potassium. Okay. Now see, whenever the ATP is producing, whenever we are getting the enough amount of the ATP, now whatever the changes we will see in these cases. So whatever the changes we will get in these cases. Now see, whenever we are getting the ATP, now we will see the channel in this way, because now ATP will get bind in these regions. This region was empty. Now ATP will get bind over here. As a result, we will see the potassium channel that will get blocked. It will be blocked, and whenever it will be blocked, there is no chances for the potassiums to go outside of the cell. So the potassium that will come back. For example, whenever the potassium was going out, at that time it was in the resting condition. The cell was in the resting conditions. So I'm just taking, for example, the potential was around minus 70 millivolt, resting membrane potential. But whenever you are getting that the ATP is coming over here and ATP is blocking the what? ATP is blocking the potassium channel. Now that's why it is known as what? ATP sensitive potassium channel. That's why it is known as what? ATP sensitive. ATP sensitive potassium channel. Okay, it is known as ATP sensitive potassium channel. Now whenever the ATP will come over here, it will block the potassium channel. We will see there will be no efflux of the potassium. The potassium will go back to the again within the cell. As a result, we will see the potential from minus 70 millivolt, it will be around minus 40 millivolt. And whenever we are seeing the changing in the potential, at that time we will see the voltage sensitive calcium channel will be open. Now see, whenever it was around minus 70 millivolt, there was calcium channel, but this calcium channel it was off, it was closed, because there was no such voltage changes. Now whenever the voltage, okay, voltage sensitive calcium channel. Now see, whenever you are getting such changes in the voltage, at that time we'll see what from minus 70 millivolt, whenever the voltage will come around minus 40 millivolt, at that time we'll see the voltage sensitive calcium channel that will be opened. In this portion the calcium channel is not yet open, because in these conditions we are getting minus 70 millivolt. Okay, in this voltage the calcium channel is not stimulated, but whenever they will see there is changes in the voltage from minus 70 millivolt to minus 40 millivolt, in these cases we'll get what, the voltage sensitive calcium channel that will be open. And whenever the calcium will be open, there will be influx of the too much amount of the what, calcium, influx of the calcium. Now what is the function of the calcium? I have already said you over here, the insulin will get stored where, within the vesicle after the Golgi apparatus. Now see, for example, is the vesicle, where insulin is stored. Is the vesicle where insulin is stored, just like this, and C-peptide. Okay, now what's the function for the calcium? This calcium will come over here, and this calcium will bring the vesicle stored with the insulin, where within the, they will get fused. That means the membrane, okay, the membrane of the what, insulin vesicles over here, okay, will get over here, that will get attached with the membrane of the pancreatic beta cells. After that we will see, after the fusions of the membrane we will see, there will be formations of the also what, output just like this. Okay, now the insulin, okay, the insulin and the C-peptide, that will be secreted out. So we are getting over here the insulin and the C-peptide. Now in this region, after that we will see the insulin that will be secreted over here. So this is all about how the insulin is getting stored over here first, and how the insulin is coming outside of the cell. That means the section of the insulin. Okay. So these are the mechanisms. In the next part of the video, we'll talk about how, what are the drugs that can affect in the different steps in terms of the diabetes. Okay. So if you think that video is helpful for you, please do like and subscribe my channel, and try to share among your friends. Okay. And see you in the next video. Thank you for watching.